Mr. Sudhakar Malla from the department to welcome all. Over to you, Mr. Sudhakar Malla.
Am I audible?
Yes, sir, it is audible. Yes, you are.
Yeah. Buongiorno, Alessandra.
Buongiorno.
Sono io, Sudhakar. Come stai?
Very well, thank you. You've done your homework for this meeting. I'm very pleased to hear that.
Grazie mille.
Grazie.
Thanks a ton, Alessandra. Thanks a ton for accepting our invitation in a very short span of time.
It's a pleasure.
It's almost a 10 years thing, right? 2010, I left NTU. I still remember we moving with the T25 flask to biomed facility for confocal microscope.
Yes.
Nice meeting you again, Alessandra.
It's good to meet again and to talk about microscopy again. It seems that this is our connection. It's a pleasure to be here for this meeting.
Sure, thank you, thank you. Hello everyone and welcome to today's session. I would like to introduce today's speaker. Dr. Alessandra completed her PhD in cell biology and biochemistry and did a master's in biotechnology. She had immense research expertise ranging from pollen germination to cell addition to kidney fibrosis. Her work mostly confines to microscopy studies, which made her more passionate towards the lens. And she joined Nikon Instruments in 2012 and worked supporting the confocal customers across Europe. Later, she worked for 3 years at SN Biosensors Sartorius, managing the field application team for live cell analysis. And now she is heading as a business development manager at Crest Optics, Rome, Italy. And now she is in charge of Crest Optics business development globally. Thank you, Alessandra, once again. And over to you, Alessandra. The portal is yours now.
Okay, so I will share my slides.
Yes, ma'am.
Can everyone see it okay? Please let me know if you can see the slides and then I will start.
No.
No, I guess.
I will try again to start the sharing. Does it work now?
Yeah. Now it's fine. We can see the screen.
So you can see okay the screen. Okay, perfect. So thank you again for inviting me to host this short seminar about microscopy. As Sudhakar very kindly summarized, I've been working with microscopy for many years. It's about 20 years now. And throughout all my career from research to industry, microscopy has always been a very strong interest of mine. So I hope with this seminar, I can introduce to you some advantages that microscopy will bring to your research and explain to you how to pick the best technique and the best specifications for your work. So this is the agenda that we will follow today. The presentation will be shared at the end of the session, so you do not need to copy all the details. And you will see that at the end of the presentation, I have also listed some resources that you can reference to find more information. Many of the topics that will be treated today, they will need each of them a one-day seminar. So of course, some topics we will just briefly touch so that you know that they exist. We can, of course, discuss if any follow-up is needed. I will not talk to you from a optics or physics point of view. My background, as Sudhakar said, is in biology, biochemistry, biotechnology. So I am more on your side than on the physicist's side. So forgive me if I am not as technical. I will be more focused on applications. So today we will go through the main features such as resolution, speed, field of view, what they mean, what they give to you in microscopy, and then some tricks that you can keep in mind when you select the best instrument to use for your research. And we will have a short introduction to super resolution as well. So I want to start with why imaging, why microscopy are so relevant, so important in research. And I'm a strong believer of seeing is believing. So, differently from other techniques in research that appear and disappear very fast, or appear, they reach a certain level of development and then they stay like that for the future, microscopy is a technique that started at the end of 1600. This you can see on the left, the first microscope invented by van Leeuwenhoek in the Netherlands. And, from that time until now, microscopy not only still exists, but it's been in continuous evolution. So if you look at the microscopes and at the images that were acquired years ago, of course, 400 years ago, images could not be acquired. Humans could only look at samples through a rudimental microscope and make a drawing manually. Then cameras and detectors were invented, so you could record what your eyes would see to make it more shareable with other people. And now we have technology that allows to record in lifetime very highly detailed images, so you can share your data in a more reliable way. And the evolution doesn't stop here. So every year, every month, there is new research, new techniques being invented, and the instruments themselves, together with their software, are getting more and more sophisticated every year. So it's very interesting, and I'm very passionate to work in this field. And I hope I can share some of this passion with you. Now, why imaging? Why microscopy is so popular? There is so many techniques out there. So why microscopy among them? And here I present a small example, which I'm sure most of you can relate to. So let's just look at studies of protein expression. I'm sure that many of you, or maybe all of you, are familiar with Western blotting, a technique where you make a cell or a tissue extract, you separate the different components, and then using an antibody, you go and identify if a protein is present or not in a sample. So the typical result is the image that you see there on the bottom left. And this is a Western blot to analyze the presence of tubulin. I could analyze the same protein by using microscopy. So I could mark tubulin in my cell and reveal it with a fluorescent antibody. So those two experiments, they point out at a very similar results, but with a completely different approach. So what do I get from them? Of course, if I look at my Western blot, I get to know if the protein is present or not in my sample, and I can do a quantification. Mostly I can quantify it as a ratio compared to other markers in my cells. This can also be done with immunofluorescence, so I can quantify per area or per number of cells how much of a certain protein I have, but I can also have additional advantages. So first of all, I can look by eye and see where exactly in my cell the protein is located, while on a Western blot I have a band that doesn't tell me where exactly my protein is unless I have made a subcellular preparation of my cell homogenate. Also, with fluorescence, I can see if this specific protein is interacting with other markers in my cell. I can do a live imaging, so I can check how the expression and the localization are changing over time. And last but not least, I get an image that is very nice to look at. And of course, our eyes are important in deciding what is good and what not. So it is good as a side effect to have good-looking data. So immunohistochemistry or immunocytochemistry are only one example, but there are many techniques where microscopy can bring advantages. So first of all, you can look at tagged proteins over time. So you can do imaging on live cells. You can analyze interaction between protein and receptors. You can check the 3D structure, because biology does not happen in 2D, but in 3D, in cells and tissues. You can analyze transcriptomics, so where a certain RNA is present in your cells or tissue. You can analyze activation of cells. This is very popular in brain research, where using calcium signals, you can check which neurons are firing, which one are active. And also you can do some behavioral studies on small organisms. This is quite popular, for example, with C. elegans. So there is a variety of studies that can be done through microscopy. And this is also very clear if you look at statistics on PubMed about published results. So since microscopy became an established technique in research laboratories, so since the images could be not only seen, but recorded, almost all published papers contain data that are generated through microscopy. And as you see from the graphs, the trend is still increasing very fast. Together with that, of course, you know that most research laboratories, if not all, have one or more type of microscopes that are present in the institute. And what I want to do with this presentation is guide you through what is important to know about those microscopes, because of course there are very big differences between them, and each of them is good for a certain application. So what we will look at today is what really matters when you want to do high resolution imaging. So in particular, we will look at resolution, especially with 3D imaging, speed, and field of view. So here, I will touch the concept of resolution without going into the physics, and I hope this will be clear enough. So resolution is a very simple concept. Basically, the resolution of a microscope is the shortest distance between 2 points that you can clearly distinguish between each other. So if you look at the image on the left, this is an image of objects that are not resolved. You cannot say if this group of dots is one object or many objects located together. However, if you look on the right, you see the same type of sample at high resolution. So you can actually see that we're looking at different dots and they are spatially separated. So this concept is behind the definition of a high-resolution microscope. So a microscope that allows you to see fine details in your samples. And the main factor that influences the resolution is the numerical aperture. This is a value, numerical aperture, or NA, is a value that is indicated in every objective that you might use in microscopy, no matter which brand it comes from, if it is Nikon, Olympus, Zeiss, Leica. Each objective, together with the magnification, will show a value that is numerical aperture. This is a value that goes from 0, which means basically no resolution, until 1.5 for the very high-end objectives. So here, and this is the only physics part, if you see, again, the same image that we saw before on the left, low resolution image, this is what you will have if you use a low numerical aperture objective. And the reason for this low resolution is that if you imagine an individual point, so a 3D individual point, looked through this type of microscope, the signal from this point will look very spread in space. On the other side, if you have a high numerical aperture objective, so here we're talking about something that has, in this case, numerical aperture of 1.3, you see that the same object will
আমি
appear
গুগল মিটে
with a very narrow signal. So this means that if you have two of these signals close to each other, as they are very narrow, you can distinguish between them. This is the base of high-resolution microscopy. So, for example, if you have to select which objective to use on your microscope, it's not only the magnification that will give you a good or a bad image, but it's this numerical aperture value. So, for example, you might have the choice between two different objectives that are 60x magnification. But if you see here, the one on the top has a NA of 0.7, the one on the bottom has 1.4. So definitely the one at the bottom would be the best choice if you want to do high resolution imaging with your confocal. The images will be more neat, you will see finer details compared to the objective on the top, which is good for other type of applications. Now, of course, this resolution discussion is valid for 2D, so the ability of distinguish points that are next to each other. But of course, biology happens in 3D. So it is also important to be able to spatially resolve objects that are in different focal planes, so that are located in the Z axis, because this will allow you to have structural information about your sample, to know exactly where a protein is localized. If you think about a cell, you could have a nucleus and a protein that is located on top of the nucleus or at the bottom. If you cannot do optical sectioning, you will not know where this protein is located. And very important, if you can do optical sectioning, so if you can slice through your sample in Z, you can also be much more accurate if you want to analyze colocalization of proteins, because you will know if their signal colocalizes in X, Y, and Z. So what do you need for this optical sectioning? Here is where the concept of pinhole, which some of you might have heard before if they have done confocal, becomes important. So let's look for a moment at the drawing on the left. Here you see the typical representation of a wide field microscope, sometimes also called the fluorescent microscope or epifluorescent microscope. In this case, what happens is that you have a sample. So here it is represented by this rectangular drawing at the bottom. Your whole sample is illuminated by light, and then light coming back from this sample is entirely collected by a detector, which is typically a camera. So signal coming from all focal planes is collected in one single image, without discrimination between focal plane. You see here that the focal plane of interest is represented by a dotted line, but we are illuminating and detecting all the sample, not only that dotted line. On the other side, if we move through a confocal microscope, we use this pinhole, basically just a hole in a membrane, to discriminate between different focal planes. So if you look at the image here, you see that through the light source, our illumination is not spread across the whole sample, but is focused on our focal plane of interest. And the same happens with the detection. So blue line is our excitation. You see we are exciting with the cone that is centered on a specific focal plane. And the green light, which represents the signal coming from the sample, is collected specifically only from this focal plane. All the light coming from out of focus signal, so above and below this focal plane of interest, is rejected. You see on those small images that this light will not enter the pinhole. So with this technique, we can image an individual point in X, Y, Z coordinate inside a cell or a tissue. Of course, we do not only want to image a point, we want to image a wide area. So what is done in confocal is that you move this point in XY, and that allows you to get a full image of your tissue or of your cell. And translated into pictures, you can actually see how the same tissue, in this case a mouse kidney tissue, will look like if you do wide field illumination or confocal. So you see that in wide field, we have a lot of out of focus contribution to light. So you see that the image is a little bit blurry. On the other side, if you do confocal on the same sample, you get a very sharp, well-resolved image, because you are, through the illumination and detection, you are looking just at a specific slice in Z in your sample. And of course, what you can do is that you can slice your sample in many different components, so you get to see very important structural information. Here for you, and I know that there are some students from botanics studies, so you will relate to this type of image. You see how a pollen grain would look if you do wide field microscopy, so it will just look as what you see on the left. However, if you can do optical sectioning, you can distinguish between the outer shell, the inner components, and you can really analyze in detail the structure. Okay, so let's talk about speed for a moment, because all these are good-looking images, but let's see how long it takes to generate those images, because that is very important. So with this point scanning confocal that I mentioned to you, of course, we have a huge increase in resolution, but we are looking at one point at a time. So this individual point needs to be raster scanned through the sample to obtain a full frame. And then we have to move in Z to get frame by frame our optical sectioning. So typically, each of these images, so each focal plane, takes about 1 second, which might sound fast, but in certain applications, this might not be enough. And I refer to applications where, look at this sample here, we're looking at trafficking of some proteins into a plant tissue. These are dynamics that are happening in milliseconds. So you need to take an image every about 10 milliseconds to be able to see this fast movement of proteins. So you will clearly understand that if my system can only acquire 1 picture per second, I will not be able to get this type of results. On the other side, speed is also important for new techniques that are being developed. Now, tissue clearing, a technique that allows to make biological tissues completely transparent, so easy to image through a microscope, is becoming very popular. With this technique, you obtain tissues that are several millimeters thick. We're not talking about a cell monolayer, but about a whole organism or a whole organ. So what you can do is that you can make a slice of this, and then you can image in 3D to get a nice structure. But of course, if you want to image a very thick tissue, as you see in the example here on the right, you need to take many images in Z to have a good resolution, so a good looking image. In this example, we need 1,000 images in Z to get this type of 3D results. So of course, if you are using a slow instrument, this will take several minutes or even hours, depending on the size of the tissue that you want to image. So there is a way to increase this speed. If you remember the drawing of the system, the confocal system with the pinhole, that one has some limitations. So what I want to show you is how you can get higher speed by using a different illumination, and then of course using optimal detection and a large field of view. This will allow you not only to get better temporal resolution from your live samples, but also to be faster when you acquire big samples. So if you can be faster, of course, you can generate more results, you can be more productive, and if you have to pay to use an imaging facility, you will have to pay less. Or with the same amount of money, you will get more work done. So that's a win-win situation. So if you remember what I said to you about the standard confocal, you have this beam of light, and point by point, you have to move through the specimen. So pixel by pixel, you generate an image. Some very smart people about 15 years ago had a good idea of, instead of using 1 point per time, so 1 pinhole at a time, they had this idea of generating a disk that contains thousands of pinholes, make that disk rotating very fast on top of a sample, so that at the same time you can illuminate the sample in multiple positions using these moving pinholes. And this allows you to acquire not an image pixel by pixel, but a wide area. So you have your 1,000 by 1,000 pixels in one shot. And you can increase the speed significantly. So typically you will go 10 to 100 times faster compared to the standard confocal. So this, of course, will have a variety of advantages. So let's just summarize here the 3 techniques that we've looked at so far. In the center, you see wide field illumination, your standard fluorescent microscopy where you do excitation and detection of everything in Z. With laser scanning confocal that you see on the left, you see that point by point, you can analyze your images in separate focal planes. So you will have a nice 3D reconstruction, but it will be slow. On the right, you see the second approach to confocal, the fast confocal, where your individual focal planes are acquired as one image, so fast acquisition. And again, you can do Z sectioning with spatial separation. So this will be extremely fast. So here I want to give you some examples of these speed and disadvantages. So which new discoveries can you do? Here you see 2 examples of thick 3D samples that require confocal microscopy to be imaged in details. So you have a parametrium on the left, and on the right you have this example of biomaterials. So these are nanotubes filled with a fluorescent solution. Those are, I refer to the biomaterials, those are about 150 micrometers thick. So for microscopy, 150 micrometers is a pretty thick level. To image a structure like this with a standard confocal microscope, with a laser scanning confocal, it will take you about half an hour. With a spinning disk, we can do this acquisition in less than 1 minute. So that means that we can analyze dynamics in a very big structure. We can analyze something that is happening fast, while if it takes half an hour or 1 hour for the acquisition, you lose any event that is happening over time. Another example where having a fast confocal technique enables you to have new discoveries is this example here, where we are looking at a slice from a brain organoid. And here, not only we can see fine details, so high resolution, but we can see 3 different channels that are very well separated in the spectral range. So we have DAPI with blue, GFP here in green, and we have near infrared with this magenta staining. So with spinning disk confocal, you can image all the areas of the spectrum because you have very high sensitivity from UV to near infrared. This is hardly possible with standard confocal because you cannot detect signal in near infrared. Other examples that I want to show you where speed and resolution are very important. This is a mouse embryo. You see from the scale, I'm talking about a very large sample. This is about 40 by 15 millimeters, not micrometers. If you do have a fast confocal system, like a spinning disk, you can image this type of sample at very high resolution fast. If you have to do the same example, the same sample with a old-fashioned or standard confocal, again, this will take you several minutes or hours. So you can have less data done in the same time. The last example, I've already shown you this image, but I want to show you, because we've done some work with this specific researcher. She had issues because she wanted to image this mammary gland tissue, but it was taking her from a single field of view about 20 minutes to do imaging in 3D with her laser scanning confocal. By providing a spinning disk to this researcher, she went down to less than 1 minute for this type of imaging. So now she can analyze much bigger areas of tissues in a faster time and a higher resolution. So these are all new discoveries that can be enabled if you do have the right equipment. So here, I do not want to spend more time on this, but I will leave you this link because we really like to look at new applications with the researchers that we collaborate with. So on our website, you will see a series of application notes describing how you can use microscopy on different type of samples. So if you are interested, just go and have a look and check the new material that is being released. Now, we have discussed about resolution, speed. Next point I want to touch is field of view. So this is how big is the area in your sample that you can image in one shot. Because of course, you might want to image just one single cell, so you don't need a large field of view. But more and more, it is important to get data from many cells, or maybe you want to look at organoids, tissues, small organisms, so large samples. And then having a large field of view becomes extremely important. So here in this image, you see field of view, so the area on, in this case, a cellular sample that you can image with different instruments. We start from the smallest, the red square in the center. So this is typically how much you see if you image with a point scanning confocal with a 60x or a 100x objective. So typically you see one cell or a fraction of a cell, depending on the cell type that you are looking at. So of course you get very high resolution, very good details, but you are limited to one cell. So you need to collect many images to get more results. When you move to spinning disk, automatically the field increases. So you can have standard field of view spinning disks, where the field of view is indicated in yellow here. So you see that you are already about 9 times bigger compared to the standard point scanning confocal. There are also versions of spinning disk that allow you to do large field of view. So in this case, we are talking about 10 to 12 times bigger compared to the standard field of view confocal. So if you then compare the speed, where it takes 1 second to acquire this red square and 100 milliseconds to acquire this green square, you will understand the advantages in speed and field of view that are combined together. Field of view is, of course, very important. We want it to be as big as possible, but to get relevant images out of your field of view, you need it to be homogeneously illuminated. So typically, if you take a light source for microscopy, you have an illumination profile that is a beam more intense in the center and then more shallow at the borders. So if you illuminate a sample with a light like this, you will have stronger signal from the center of your sample, dimmer signal from the borders. Maybe some of you have seen these tools devices that make sure you have a uniform illumination. So you can modify the shape of your light to get uniform illumination. This is, you're looking at one point, you're looking at a wide area simultaneously. You want to have all that area that is uniformly illuminated. So together with the light source, also important to look again at the specifications of your objectives. So again, all brands of microscope use the same coding. Plan means that the light that is going through the objective is homogeneously distributed when the sample. And the animation will give you the best imaging results. And here I want to show you the effect of these homogeneous illumination. So we're looking at a cell monolayer, large field of view illumination, so we can see many resolved, but the image on the left is obtained without uniform illumination. So you actually see very signal for the cells in the center, but at the borders, the image becomes quantification of the cell that is at the center of this image with the one of the cell that is near the borders, because I know that my signal is not influenced by the expression, but is influenced by the intensity of my illumination. On the other side, if you look at the image on the right, you see that cells across the whole field of view have the same type of signal because they are illuminated with a uniform light. So with this type of image, not only I can collect data from tens of cells at once, but I can also confidently compare signal of one cell in the center with one cell at the borders. And with confidence, I can say this cell expresses more protein than the other cell, because all the other factors that can influence the fluorescence are taken care of. The other big advantage of having a homogeneous illumination comes when you want to image a large sample that requires a collage to generate a final image, so not one individual picture. So if my image is not uniform and I have these dark borders and corners, when I try and do a collage or a stitched image, I will have these black squares. So these are the borders of the individual pictures that it took to generate this large image. On the other side, if I'm able to have a homogeneous illumination and I have the correct objective lens, I can image a very large sample without being afraid of this dark shading. So these are some further examples for you that we have acquired in our lab. So you can see an example from mouse brain. This image here is made of about 150 small tiles stitched together. And you don't see any of this dark shading. Other examples are with organisms, in this case, C. elegans, where we look at the movement and at the activation of neurons, which in this case are labeled with GFP. So having a large field of view with uniform illumination allows us to see many, in this case, many C. elegans in one image, and to compare intensity and activation of neurons across the whole field of view. Okay, so these are what we have seen so far are the main factors that you need to keep in mind when approaching high-resolution microscopy. There are also other tricks that I want to share with you, and those are related to labeling and detecting signal in your sample. So first of all fluorescent microscopy, it doesn't matter if it is wide field, confocal, super resolution, it all starts with labeling your samples with a fluorescent signal. So this can be a fluorescent tag that is attached to your protein of interest, or it can be a fluorescently labeled antibody. It doesn't matter. Each fluorophore, or each molecule that can express fluorescence, will have an excitation and an emission peak. So you have, in this case for GFP, you have your excitation, which is done with blue light, the peak is at about 480 nanometers, and the emission is indicated in green. So the emission is always shifted towards a longer wavelength, which means less energy. So for each fluorescent signal, you have your peak of excitation and emission, and you want to match the specification of your microscope to these specific peaks. Now, if you are looking at one single fluorescent protein per sample, you don't have to worry much about what you choose in terms of fluorophore. You can choose the ones that you have in the freezer in terms of reagents, or match the excitation lines that you have in your microscope. However, if you want to look at multiple fluorescent signals in your sample, you have to be a little bit more careful on the fluorescence that you choose. And especially, you want to be careful not having overlap of these emission signal, because you will get data that are kind of contaminated by different proteins at once. So for example, if I wanted to choose 3 different labeling, 3 different fluorophores for my sample, in this case, I might want to choose something that is on the blue, something on the green, something on the red. If I select yellow, orange, and red, you see that those peaks have a lot of overlap. So when I go and detect my signal, I will detect, even if I have 3 separate antibodies for 3 separate proteins, I will see all the signal overlapping. I will not be able to tell what is what. So always be careful when you select your antibodies to have the best possible separation in the spectra. So this will allow you to do applications like this one here, this beautiful rainbow image, where you have a variety of fluorophores from blue to far red. If you don't have a good spectral separation between your fluorescent, all of these will look at the same color. You will not be able to distinguish between cells. The other tricky consideration is about autofluorescence. Most tissues, especially plant tissue, but also some mammalian tissues, they have autofluorescence. It means that if you look at them under a microscope, even without doing any staining, they will have some bright light emitted from them. This is particularly true with plant, and for those of you like me that come from plant biology, we all had big headaches when trying to do microscopy on samples like this, because the sample already is very bright. So sometimes you can take advantage of this autofluorescence, because you can do imaging without treating your samples. So if you want to do structural studies for pollen, for example, you can take advantage of the autofluorescence, put your pollen under a confocal microscope, and get a beautiful image. On the other side, if you need to look at specific markers, you want to make sure that you know in which color your sample is fluorescent, so that you select markers that are labeled with a fluorophore far away from that color. So again, we go back to this choice. If you know that you have autofluorescence in the green and yellow, you might want to choose red or far red for labeling your antibody, so that you don't have any interference. The next bit is about light sources used for microscopy. There is, in the market, there is a variety of light sources from many different brands. You can choose typically halogen lamps, LEDs, lasers. The most popular nowadays are LEDs and lasers. And here, I just want to leave a reference table for your consideration. Main differences between LEDs and lasers. So if you look at the typical LED light source indicated on the top, this light source will give you light from UV to far red, typically. So you have many different lights, meaning that you can work with different fluorophores. Those solutions are cost-effective. They last for a very long time. They are stable for a very long time. So we're talking about typically 20,000 hours, so very long duration. The disadvantages is that if you look at those excitation lights here, so those bands, they are pretty wide. It means that, for example, if I want to do excitation of my GFP, where I know that I need the light at 488 nanometers, an LED will give me 20 nanometers before and after that. So I will also get some aspecific signal from my sample, because I might get excitation of some autofluorescence or of some other labeled structures. On the other side, if I select a laser-based light source, you see a drawing here at the bottom, I can have the same advantages in terms of number of lines, but those bands are very narrow. So I can be more specific in what I go and excite in my sample. So I can have better specificity of my signal. So again, I can increase the resolution because I minimize background and autofluorescence. Of course, there are differences in cost. Typically, a laser is more expensive than an LED, even though with modern technologies, lasers are becoming very approachable as well. So in the past, there was maybe an order of magnitude in price. Now, many of these sources are kind of aligned in terms of pricing. The other advantage that laser lights will have compared to LED is that typically they have higher power. So it means that you can excite dimmer signals. So you can look at more samples compared to what you can do with an LED. The last bit about equipment is the detector. So normally when you do confocal spinning disk or wide field microscopy, you will use a camera for detecting the signal. There are hundreds of different cameras available on the market with many different specifications. The important bit for you to consider is the quantum efficiency of the camera. Quantum efficiency simply means how much light coming from the sample is translated into my digital image. So how many photons I need from my sample to get a good picture. Modern cameras, and there's been a huge evolution in the last 5 to 10 years in this world, modern cameras are able to convert 95% of signal coming from the sample in your digital image. So basically, you don't waste any light. Cameras that were used 10 years ago, they could only convert 10% of the signal from the sample. So you needed much stronger labeling, much stronger excitation light, but that had the drawback of bleaching the samples. So now technology has done a huge progress on this field. Of course, when you select a camera, and this I don't want to go too much into details, but you also need to check the signal, so the quantum efficiency, but the pixel size as well. So cameras with very big pixels will give you good signal, but lower resolution. So your images will be more pixelated. Cameras with small pixels will give you better resolution because you divide your image in many small squares. However, as each pixel will be smaller, it will collect less light. So the images might be a bit dimmer. So it really depends on the sample type. But here, camera makers they offer a lot of tutorials on the best camera for each application. So if you are interested, just have a look at some of those resources. So this is it for wide field, and I would call it standard high resolution microscopy. I just want to give you a very brief introduction to super resolution. Here, a good coverage of super resolution would take a 1-week seminar. So we are just taking 5 minutes. So this is a very condensed version of it. But basically, we started the presentation talking about resolution. And confocal is the instrument that can give you the best resolution that physics allows. However, in the last 10 years, techniques have emerged that can go beyond the limits of physics, using some artifacts in illumination and in data processing, and also some techniques that take advantage of chemistry of fluorophores. So here you can see a scale of living organisms from large to very small. And you see that with wide field and confocal microscopy that we have discussed until now, we can cover until a resolution typically of 200, 250 nanometers. So we can go down to this level. We can see some subcellular structures in details, but we cannot see other smaller structures. Viruses, for example, something that is very popular right now, but also smaller subcellular structures or individual proteins. So in that case, we go below 100 nanometers range. So you see that there is a variety of techniques that allow you to go lower in resolution, so higher in resolution, which means looking at smaller objects. And there is a continuous evolution of this. So super resolution emerged about 10 years ago. There were at that point only 2 techniques. Now there is about 50 different techniques grouped into different types. So there is a whole world of super resolution. Today, I will just give you a short overview of structured illumination, which you might have heard as SIM, structured illumination microscopy. Sometimes it has other names, but it is basically a technique that allows you to double the resolution compared to your confocal microscope. So as I said to you, with confocal, you go to about 200, 250 nanometers resolution. With structured illumination, you go down to 110, 120 nanometers. So you can start looking at structures like mitochondria, viruses, and things that are in this order of size. Now, structured illumination. It's different from what we've seen until now, in the sense that you don't get a direct image of your sample, but there is some special illumination and computation involved in the process. So until now, we've seen an illumination of the whole sample with a nice and uniform light. Forget about this. What you do with structured illumination is that you illuminate your sample through a pattern. So in this case, we see a pattern made of different dots, but those could be slits of cut inside a matrix. So basically, when you illuminate your sample, in this case, we have a cell, it will look like this. Light will only go through these dots here, through these holes. So what happens is that this, that is called a mask of illumination, so these dots are moved in X and Y over the sample until you get a group of images that are then recombined together to give a high resolution image. There is a lot of physics and elaboration going on behind this, but the final results is what you can see in those images. So I go back to a kidney sample, which I'm very familiar with, and Professor Sudhakar is also very familiar with from our work at Nottingham Trent University. So here is the same sample seen in wide field, in confocal, where we can see optical sectioning, so less blurred image. And here is what you would see with structured illumination. So all the structures, the labeling here looks thinner. You have dimensions that are closer to the real dimensions of the structures. So again, what new discoveries come out from this? Look at this example here. We're looking at neurons where we have green microtubules, and in